Hello, thank you for joining me. My name is Shady Kuster, and I'm a senior in the Presidential Honors Program here at Tarleton State University. And today I will be presenting my research over the conformation of extreme mitonuclear discordance of the pocket gopher. Because that title is kind of a lot, I'm going to break it up into some pieces. That first piece is going to be the pocket gopher. The particular species I worked with was Geomys breviceps, or Baird's pocket gopher. And pocket gophers are small burrowing rodents that live in sandy soils. They're distributed across Texas, Oklahoma, Arkansas, and Louisiana. They're called pocket gophers because they have these external cheek pouches where they can store food items for later consumption. And these go pretty far down their bodies, and you can actually invert them just like you can your pants pockets. And so that next bite-sized piece is going to be mitonuclear discordance. And this is just some sort of disagreement between something to do with the mitochondria and something to do with the nucleus. And so our cells actually have two genomes within them. One is within the mitochondria and the other is within the nucleus. And so those are called respectively the mitochondrial genome and the nuclear genome. Theoretically, since they are within the same cell, which make up the same organism and are part of the same population and etc., they should show similar evolutionary patterns if there's nothing weird or strange going on. Baird's pocket gopher, however, does not show a normal evolutionary pattern. Instead, we have this discordance between the mitochondrial and nuclear genomes. In the mitochondria, we've developed this data set that shows 4 divergent lineages. Here on the right is what we call a phylogenetic tree, and this is created during phylogenetic analysis, where we are trying to see how distantly related two sequences are from one another. So a longer branch length indicates that two sequences are more distantly related. And so we see these 4 groupings here, which show the 4 lineages. And this picture here on the left just shows the geographic distribution of Geomys breviceps, and the dots are colored to match the 4 lineages shown here on the right. In the nuclear genome, we performed a principal coordinate analysis right here, shown here on the right. And you can see that these dots are colored to match the mitochondrial lineages shown in the previous slide, but they don't group isolated from one another. If they were to show the same pattern as in the mitochondria, you would see a group of reds by themselves, a group of yellows, a group of greens, a group of blues. They would be in their own individual groups. But we see here that the green, yellow, and blue has overlap, and even to some extent overlaps with the red as well. And so because they fall out as they do geographically with the red on the bottom, the green, reds, and blues in the middle, and then the blues further up, we call this isolation by distance, where these sequences are distributed across a geographical pattern. Something else that we needed to take into consideration since we were working with mitochondrial sequences is something called Pneumites, or nuclear sequences of mitochondrial origin. This occurs whenever a sequence from the mitochondrial genome becomes inserted into the nuclear genome by one mechanism or another. This can cause problems with genetic studies using mitochondrial sequences, because if you're just trying to look at a mitochondrial sequence and you have something that is located elsewhere but is something that is just slightly different, then it's a sort of imposter, and it can really confound that data set and can lead to some erroneous conclusions. And so I wanted to take that into consideration as I was doing this analysis and trying to validate these 4 mitochondrial lineages. There's a very strong discordance between the mitochondrial and nuclear genes, and we have seen evidence of Pneumites in some of our samples, so we needed to be aware of that. And so in order to confirm that that mitochondrial data set was indeed made of mitochondrial sequences and not Pneumite sequences, I needed to validate those 4 divergent mitochondrial lineages by ruling out the possibility of Pneumite contamination. And I did that by sequencing entire mitogenomes from a representative of each of those 4 lineages while avoiding Pneumite sequences using methods that would prevent those Pneumite sequences from being a part of the analysis and a part of the data. Once I had those mitogenomes, I compared them to previous mitochondrial gene sequences, those used to make the tree shown previously, and then perform a new phylogenetic analysis using those mitochondrial genome sequences. And as I generated these mitogenomes, I wanted to make sure that despite our efforts to prevent Pneumites from being included in the data, that was indeed happening, that we were actually working with mitochondrial sequences. And so here you can see that I performed a phylogenetic analysis, which has created a tree. And this sequence down here from the methods that we used did not group with the Pneumite sequences. And so that tells us that this sequence is of mitochondrial origin and is not from the nucleus. So then we could continue on with sequencing all 4 lineages. And once I had those genomes put together, I could then perform this phylogenetic analysis to produce this tree. And they fell out where they were supposed to. So they fell into the lineage that I took them from. And this proves that the original mitochondrial data set was showing an evolutionary pattern based on mitochondrial sequences and did not include Pneumite sequences. And so I was able to validate those 4 lineages. But we still have this weird pattern of evolution between the mitochondria and nucleus. And one possible explanation for that is something called mitonuclear coevolution. This is something that happens over a very long period of time whenever you have 2 genomes interacting with one another. And in this particular instance, mitochondrial genes become dependent on nuclear genes for their functionality. And so if you have a cell like this one here on the left, where the mitochondria is made up of genes that are dependent on yellow lineage associated nuclear proteins or genes, then you need yellow lineage associated genes in the nucleus in order for that mitochondria to be functional. And so the cell on the left is good because it does have those yellow genes in it. However, if you have a cell here on the right where the mitochondria still needs those yellow lineage associated genes, but there's none of those in the nucleus, that cell and that offspring from that cross between gophers is not going to develop, and you will not see that in the data set. And so this will produce what appears to be 4 highly divergent mitochondrial lineages, while the nuclear data will show just simple isolation by distance because those genes can be crossed. And so then I wanted to look more into substitution rates as another possible explanation. And a substitution is just a single change in DNA from one nucleotide to another, so just one letter change. And there are 2 questions that I would like to investigate. The first being, do mitochondrial genomes in Geomys breviceps have a higher substitution rate when compared with nuclear genomes of Geomys breviceps? And the second is, does Geomys breviceps have a higher mitochondrial substitution rate when compared with other species of Geomys? Because we don't see this pattern in any other species of Geomys. I'm currently still working on generating data to answer these 2 questions, but hopefully that will come to a conclusion here in the next couple of weeks. Overall, I've really enjoyed working on this research project. It has really led me to pursue a career in research, particularly in genetics and evolution, because so much of genetics now relies on computation. I did pursue a computer science minor, which has helped me shift my focus more onto the computational biology side of research. I'm very excited to attend Colorado State University in the fall, where I will be attending their grad school to further my research career. And so I just want to thank any of you guys for watching. If you have any questions, feel free to reach out, but thank you very much.